Good afternoon, everyone. We'll get started in just about 1 minute. We want to allow everyone ample time to filter in from the waiting room, but thank you for joining us. And we'll get started in just about 1 minute. For those of you just joining us, we're waiting about another 30 seconds to get started. We're letting everyone come in from the waiting room, and it's great to see so many familiar and unfamiliar names joining us. All right, let's go ahead and get started. Good afternoon, everyone, and welcome to the IMSMP and Tisch MS Research Center of New York's Quarterly Connect webinar series. I am so glad that you've joined us for an introduction to the science of organoids. They're small in size, but they have big implications for MS research. For those that don't know, Tisch MSRCNY is an independent nonprofit research center dedicated to finding the cause of and cure for multiple sclerosis. We're located in the heart of New York City, where we conduct industry-leading medical research that rapidly translates from the lab bench to bedside. That translation happens because Tisch MSRCNY collaborates with the International Multiple Sclerosis Management Practice, a world-renowned clinic that offers MS patients access to the best available treatments and integrated support services. Together, Tisch MS Research Center of New York and IMSMP make up the world's largest MS center. And we are committed to expanding the scientific understanding of MS while redefining patient education and involvement in research, which is why we offer webinars like these. We truly want you to know what's happening behind the scenes. I'm Sarah Bond, and I lead communications for the center. I'm joined today by two scientists from the Tisch lab, Dr. Kriti Kalpana and Dr. Violaine Harris. Dr. Kalpana is a research assistant focused on, Dr. Kalpana is a research associate. She is focused on the phenotypic and functional differences across MS subtypes. Her work at the center involves leveraging 3D neuroimmunocytoid cultures to explore the role of glial cells in MS and identify potential functional biomarkers of the disease. And if that makes no sense to you, that's what we're here to talk about. So you're in the right place. And Dr. Harris is the associate director of the lab and a senior research scientist who has led two groundbreaking trials at the Tisch MS lab. In fact, they were the first ever to test bone marrow-derived neural progenitors in patients with MS. Her work focuses on developing cell therapy strategies to promote repair and regeneration. And she also leads the laboratory staff and mentorship programs. So to put it simply, she has a bird's eye view on all the work that we do, including, of course, her own, and helps connect the dots for maximum cross-functional impact. Now, today's webinar is being recorded and will be sent to you on Monday. So if you'd like to watch it again or share it with friends or colleagues, please feel free to do so. We do have a Q&A portion of today's session, and due to time constraints, we're prioritizing questions that were pre-submitted via the Q&A form that was listed on the registration page. But if you have an emergent question that was not submitted ahead of time, feel free to use the Q&A feature within Zoom, and we may not be able to answer it during today's session, but it will help inform future content from our lab. So without further ado, I'm going to share my screen for everyone, and let's get started with Dr. Kalpana and a pretty basic question. What is an organoid?
Thank you, Sarah, for that introduction, and thanks to everyone for joining in today to learn more about the organoids and the work that we do here at Tisch Multiple Sclerosis Research Center using organoid models. So to start things off, an organoid is basically a personalized three-dimensional structure made of different cells that would mimic a human organ. So these are personalized models of an individual's organs. Um, and they're small enough, but then they would have different cells that would be part of an organ. So you can look at them under the microscope and you can do research on these models.
Okay, I guess that makes basic sense. So organs are in the body. Organoids are tiny models of organs that live outside the body. Got it.
Yes, so there are several different types of organoids. Obviously, just like the different organs in the human body, we have organoids that can be made to mimic different organs. So we have organoids for looking at heart cells, which are known as cardiac organoids that you see here on the screen. And then we have organoids that would mimic the gut or the intestine, known as gut organoids. We also have retinal organoids that we can look at eye functions and eye disorders with. We have pancreatic organoids that have been used for diabetes research. And then we also have brain organoids, which is what we are using in our labs. So brain organoids or cerebral organoids are basically the model that we use for our research here.
Okay. So why brain organoids? Why are they so important to MS research? Like, why do we need them?
Yes, of course. So the way we study the brain currently is by using different approaches. Mostly it's used, most research studies use postmortem brain tissue, which is limited. So getting access to postmortem brain tissue is difficult because it would only happen once a patient has passed away. We also have animal models like mice and rats that can be used for doing MS research, but multiple sclerosis in general is a human specific disease and animals don't really get that. So animal models, even though they're great to look at certain things, they fully do not recapitulate what would happen in the human disease multiple sclerosis. And then we also have cell culture, which is sort of like what we do in a lab. We can do this in 2D using 2D cells as well. So not just 3D organoids, but 2D cells. But the problem with that is that even though they are great to look at different cells, you are still looking at them separately. And then MS is a complex disease. It does not just happen involving one cell type. There are different cell types that are involved in the brain. So it's important to look for at the different cells that are important in MS together. So for that reason, to overcome these limitations, we created what we call a brain organoid model to study MS research.
Okay, I mean, that makes a lot of sense. MS is an incredibly complex disease that is rooted in the brain. So a brain that's no longer living, or the brain of a mouse, or a bunch of 2D cells in a dish, I can see how those would not be the best tools, and that using them to study the disease in humans is sort of like a square peg in a round hole, and a 3D brain organoid is a round peg, round hole. Is that right?
That's very nicely put. Yes, exactly. So we were looking for a solution, and the solution in our case is using 3D brain organoids because the models that we had currently, they didn't really work very well for us. They are good in different aspects, but still the greatest thing that brain organoids have been able to achieve is they can be individual specific. So these are personalized models that would be specific to a given individual, and they can be made from people with MS or other diseases, or healthy individuals, which makes them such good models to use for MS research. And then the other good thing and the advantage that they have over 2D cell culture is that they can more accurately mimic the structure of the brain as compared to other 2D models. So just looking at cells. Here in this image, you can see that on the left, we have the human brain with some sort of structure in it. The different colors that you see are different types of cells in the brain. And then on the right, you see a very similar image of the brain organoid. So what you're seeing here is that the brain organoid can kind of mimic the type of cells, but also the structure that you would see in the human brain cortex. So it makes them, again, really good models to look at brain architecture as well. So basically what I want to say is that 3D brain organoid models can demonstrate different interactions that some of the other models that I talked about can't, which is the benefit that we have in using 3D brain organoid models.
Yeah, that sounds incredibly unique and I imagine very useful when you're trying to mimic the reactions within an actual brain. All of this is still sort of opaque for me though. Like, how do you make them? How the heck do we make mini brain organoids? Let's get into that.
Yes, of course. So the first step that we do is getting something as simple as a blood draw from an individual. So as I mentioned, these organoid models are specific to an individual, and they would replicate or recapitulate an individual's organs, right? So we start by getting blood or skin biopsies from an individual, from, in our case, an MS patient or a healthy individual. And then we, using that blood, we get certain cells which are more like normal cells, also known as somatic cells, from these blood samples. And then these somatic cells can then, through a process known as reprogramming, be converted into this cell type known as iPSC. iPSCs, or induced pluripotent stem cells, are cells that have been converted to a more naive, to an earlier state, which makes them really good for becoming any cell type of the human body. So we can take these iPSCs and form any cells in the body. And that's how we start.
In my brain, for some reason, I'm thinking of the wild card in Uno. Like iPSCs are the wild card. They can turn into anything. They're a blank slate. So you take blood, you reprogram them into blank slate cells, and then somehow those blank slate cells become the organoid. What's that process like?
Yes. So as I mentioned, like, these iPSCs can basically form any cell type. Obviously, for our research, we want them to form brain cells. And then since we are looking at more of a 3D model, so we want them to form brain organoids. So we start with iPSCs, and then we give them different vitamins and supplements, just like how you would want your brain health to be maintained by taking different vitamins and supplements for your own health. We kind of provide different factors that would help form different cell types that are important in the brain. So we give them factors for making neurons, for example, the nerve cells. Then we give them factors for other cells that are present in the organoid or in the brain in that case. So we have glia cells like astrocytes and oligodendrocytes that are equally important in the brain, and they're equally important in multiple sclerosis. So by giving the iPSCs different cues, either physically or with supplementation with vitamins, with different factors adding to the cell culture, we can make these brain organoids.
Okay. How long does that take, the iPSC, and then you give it food, water, and time? It's an organoid. What's that time difference until we can actually start doing experiments on them?
So it is a long process. It does take a few months, starting from iPSCs to getting brain organoids that we use for research here. We would take about 2 months. We would get organoids that are ready to be used for research within 1 to 2 months. So it is a long process, but during that time, something or the other is always happening in the organoid because you are getting all of these cell types that are important for us to look at. So we want the cells to be mature enough so we can do further research on them, which is why it does take longer.
Okay. And what about the normal cell to iPSC blank slate cell timeline? What's that like? That reprogramming process? How long does it take?
Yes. So the reprogramming process also would take about 3 months. So starting from the blood draw from an individual to getting iPSCs that can be used for making these organoids, it would take about 3 to 4 months. It is a slow process, but the great thing about iPSCs is that they have a renewable they are a renewable source, so we can keep on using them for a really long time. We can make stocks of these iPSCs, so we never really have to go back to the same individual and ask for blood draw again and again, which makes them really easy to use.
Great. So about 6-ish months from blood draw to an organoid that is ready for experimentation and the research process. Is that right?
That is right, yes.
Great. And you were kind enough to provide us with a few photos. Can you tell us what we're looking at here?
Yes. So in the top image, you can see an iPSC culture, which is, as I mentioned, a 2D culture. So we have all of these induced pluripotent stem cells, or stem cells for short, in that we are looking at. And here you can see a colony of a lot of cells. These would have a few 100,000 cells altogether, forming a colony. This image has been taken under the microscope. These colonies are microscopic, so you can't look at them by naked eye. But under the microscope, this is what you see, a colony full of tiny little iPSCs. And then on the bottom image, you see an organoid, again taken under the microscope, which has been formed using these iPSCs. So this is a 3D structure. It looks basically spherical, like a little ball of different cells. And because you can't really know what's going on, you only see it as a dark object under the microscope.
Excellent. And Dr. Kalpana graciously welcomed me into her lab. And unlike the iPSC colonies that you cannot see with the naked eye, I was kind of rattled that you can see the brain organoids with the naked eye. They're like the size of a pinhead. And of course, she showed me under the microscope where we were able to blow them up and look a little bit closer. But even with her just holding the dish, I could see the organoids there. And it's just kind of incredible to come face to face with this sort of research. So thank you for letting me come to see them. Zooming out a little bit, obviously cerebral or brain organoids have a lot of potential when it comes to researching multiple sclerosis, but I know that they are used for other diseases as well. Can you talk a bit about organoids in the context of neurological research?
Yes, of course. So brain organoids, they are used for a lot of different things, multiple sclerosis being one of them, which is kind of a new field still. but they've been used for neurodevelopmental disease research or disorder research, such as autism and Down syndrome. They're being used a lot for neuropsychiatric diseases such as schizophrenia and anxiety and depression. And they've also been used a lot for neurodegenerative diseases such as Alzheimer's, Parkinson's, Huntington's, as such. And then they've also been used for virology research, in which different viruses that might affect the brain can be added into the brain organoids. So viruses such as HIV, Zika, COVID virus can be added into these brain organoids to look at their effects on brain cells.
Wow, thank you so much. That's incredible. So this sort of helps me understand organoids are tiny copies of organs that live outside the body that are round pegs for round holes in terms of MS research. We grow them over the course of about 6 months from using blood drawn from a patient that we reprogram the cells of to make blank slate cells that we then nurture, give nutrients, give time, and they become one of the organoids that we showed a photo of. Is that right? Does that make sense?
Yes, that sums it up perfectly.
Great. So then I'd love to switch to gears to Dr. Harris. That is what organoids are and why we use them in MS research. But what has our research shown so far? Have any fruits born of this labor?
Hi, yeah, so I'm very pleased to be participating in this webinar today. I thought it would be a really good opportunity to talk about some of the organoid research that has come out, that has been performed at Tisch. And I wanted to talk about this study in particular because I think it's a really good example of how organoids and brain organoids can help us understand very complex neurological diseases like MS.
Great.
So,
as a layperson looking at this study, though, Dr. Harris, I sort of don't know where to start. Can you tell me where to begin tackling a piece of information like this?
right. So you always want to start with the title, and it tells you everything you need to know usually. So cerebral organoids, this is the organoid model that Dr. Kalpana just discussed. Primary progressive multiple sclerosis, this is the MS subtype that was the focus of this study. And the primary finding, which was that oligodendrocyte stem cell and oligodendrocyte differentiation was defected in the organoids as a result.
Great.
And then,
yeah,
you also, yeah, right. The authors, the author, the sequence of the authors is very important in every paper. So the first author, in this case, my colleague, Dr. Nicolas Davio, he was the scientist who really conceived of the study and oversaw the study. He would be here today talking about his own research, but he's since relocated back to France. And so I'll be talking about his work for him. So he's the first author. In the middle, you have our two research assistants who gave a lot of technical assistance to Dr. Davio. And then at the end, the last author, Dr. Sadiq, I'm sure many, if not most of you know, is the director of the research center. And he also conceived of the study along with Dr. Davio and provided funding for the study. So, so he's the last author.
That makes sense. What about all these numbers?
Yeah, so yeah, Biology Open is the journal. This happens to be an open access journal, so it can be, there's no paywall to read it. It is a periodical, so every month or so there's a new volume, or every year there's a new volume, and every month there's a new issue. And so that's what the numbers are, the issue and the volume number, to help you find the article.
Makes sense. And what about that one?
Oh, yeah, the DOI, that's a digital identifier. So that helps you, that number helps you find the article on the internet.
Oh, great. This on the left seems fairly self-explanatory. It looks like a table of contents.
Yeah, that's exactly what it is. It's the abstract is the summary of the study. The body of the study starts with an introduction to kind of put the research to set up the research and give you some background in terms of why you're doing this research. The results section will give you all of the results that you found. And then importantly, at the end, there's a discussion to take the results and put it into context of the scientific field. And lastly, the materials and methods is very important. These are the methods that were used to perform the research and allows other scientists to replicate your findings.
Great. All right, that clears up how to approach looking at published research, but we are very spoiled to have you on the call. So can you perhaps give us the gift of breaking it down for us and tell us what we found?
Yeah, so I chose one part of one of the figures in the paper to discuss, and it's, I think, a really exciting part of the paper. So what Dr. Davio did is he took blood from patients with MS, much like Kriti just described, made organoids from them. And as the organoids were growing, you can see in the top panel, those are the dark spheres that Kriti showed you earlier. He notes that early on, the organoids from primary progressive MS patients were slightly larger than the controls. And this difference was significantly different when he measured the sizes. Over time, the size of the organoids were the same. So it was really in these first few days that he saw this difference. And it's not that the brains of primary progressive patients are bigger, but it was just a difference in the organoid growth that prompted him to look more closely at the organoids. And so he did look at the different layers of the organoids. We saw that red layer and the green layer in some of the previous slides. The red layer stands for stem cells that are typical of the subventricular zone in the brain. And the green layer stands for neurons that are characteristic of the cortical plate, the cortical layer of the brain. And so when he measured the thickness of the two layers, he was able to show that this green layer, this cortical layer, was thicker in organoids from PPMS compared to controls. And this was due to increased migration of stem cells moving from the red layer and differentiating into neurons in the green layer. And so this suggested there was some kind of imbalance in this process going on in patients with primary progressive disease. And he went on to show that this was due to a protein called P21, which you might have heard about. There were also some differences in oligodendrocyte maturation in the PPMS organoids. And so this was the first study of its kind to investigate organoids in people with MS. And it was quite surprising because it implies that there may be inherited defects in primary progressive due to their genetics that contributes to neurological disability and possibly remyelination failure, which is characteristic of primary progressive disease. So it was a very exciting study.
Absolutely. And so the 3D nature of the model is what allowed us to see the migration. Is that right? And that's sort of the special sauce of organoids?
That's exactly right. We never would have discovered this had it not been for the 3D architecture of the organoid.
Wow, that's fascinating. Thank you so much. So with this being the primary finding from our last published paper, what are you seeing as like the next 1 to 3 years? What's the potential of this research?
Yeah, so one thing missing from these organoids, we've been talking about very simple models of the brain, but there's no immune system, there are no blood vessels. And so one of the first things we'd like to do is to incorporate microglia into the organoids. So microglia are the resident immune cells in the brain. They play a very important role in brain development. And we know that they contribute to progressive disease as a result of chronic neuroinflammation. So we're very interested in organoids, incorporating the organoid or in microglia and incorporating them into our organoids to better understand how they interact with other cells in the organoid. So that's one thing that Kriti will be doing. Another use of these organoids is to better understand different phenotypes and model different phenotypes in MS. So for example, cognition impairment is a very debilitating consequence of MS, and it happens in some people, but not all patients. And so we'd like to develop organoids from people with MS, with or without cognition impairment, to see if there are any inherent differences that can be detected using the organoid model. And one thing you can do is you can measure the electrical activity of organoids. So much like you would measure electrical activity in the brain using EEG, you can connect the organoids to little mini electrodes, basically, and measure the electrical activity to better understand the function of the neurons in the organoids. So that's another really exciting avenue of research that we're pursuing.
That's really neat. So increased specificity is what I'm hearing for the next 1 to 3 years. What about a 5 to 10 year time horizon? What do you see in this field moving forward that much?
Yeah, I think that these models will become more and more complex. So we will be including other immune cells, T cells, B cells that play an important role in MS, incorporating blood vessels to better model the blood-brain barrier, which is very important in disease development, making organoids, spinal cord organoids, because we know spinal cord disease is an important factor concerning disability. So just using this as a tool to better model neuroinflammation in this disease. And one thing that we're really excited about trying is understanding how viruses might interact with organoids from MS patients. So a virus like Epstein-Barr virus or EBV, we know plays a role in possibly triggering MS. And we're very interested in understanding how EBV might infect our organoids from different genetic backgrounds, from different patient populations, and how that might help us understand the trigger of MS, the cause of MS. We can also, one of the exciting potentials of organoids is that they can be personalized models. So we can make an organoid from the same patient, include their own immune cells, T cells, B cells, even their own CSF. So really making a little mini avatar of the patient in order to study that. And we'd like to use these, one day, maybe not yet, but one day we could maybe use these personalized models along with technologies like machine learning, AI, to better predict how patients will respond to therapies. So especially some of the newer therapies that should be coming out, targeting remyelination and neuroprotection. I think these personalized organoid models will be really helpful and really moving towards an era of precision neurology, which is I think really exciting. So a lot of exciting things to come. We're currently thinking of organoids for disease modeling and drug screenings, what I've been talking about, right? But I do think in the next 10 years, we may start to think of organoids in the context of a regenerative treatment. And, there are many, many hurdles to overcome before this is a reality, but it's currently being explored in other diseases and gastrointestinal disease, for example. So I don't see any reason why this couldn't be explored as a regenerative cell therapy for neurodegenerative diseases like MS. So it's exciting.
That's really exciting. And I know that both of you have attended some cross-functional conferences recently that have explored potential in different fields. It's sort of helping you stay abreast of organoid research in other disease areas. Dr. Kalpana, can you tell us a little bit about the conference that you attended recently?
Yes, of course. So In April, I attended the Garden Research Conference, which is a really popular conference that looks at different topics. The ones that the theme that I went to, that was the functional genomics and brain development in human disease. It was held in California, as I said, in April, and there were a lot of different talks focusing not just on functional genomics and brain development, but also using organoid models for looking at brain research. A lot of the talks focused on neurodevelopmental disorders such as autism and Down syndrome, as well as neuropsychiatric disorders like schizophrenia. One of the really cool things is that Garden Research Conferences, you basically are presenting unpublished data. So everything that I got to see is really new and cutting edge research that's happening all over the world with attendees from all over the world. So it's really a great place to interact with people, like-minded people who are working towards the same goals as you. And then some of the really fun things that I got to know is that these organoid models now are not just as simple as coming from one individual. You can get iPSCs, stem cells from multiple people at the same time and introduce them all into one single organoid to make chimeroid models. Yeah, so it's really cool stuff. And then you can look at you can look at different gene effects and other things like effects of treatments that you want to do by using these chimeroid models. And then you will get that same information for all the people that you use those iPSCs from. One other really cool thing was interspecies organoids and chimeroids. So not just making organoids from human iPSCs, but also from other animals like non-human primates, like chimpanzee iPSCs. And you can also make like chimp and human chimeroid models together. So the field is endless, and there's so much you can explore using organoids to study different things, and it just keeps evolving. Even though it's a newer field, it's only been around for a little over a decade now, but there's so much that we've accomplished in this decade.
That is really cool. Wow. I do want to say, when I first heard about this, I asked the question to Dr. Kalpana, is there the potential that these like mixed organoids will just keep growing? Like they'll keep developing? Are they going to outgrow the dish? And she said to me, well, is your brain going to outgrow your skull? And so the answer was no. So we're not at risk of like Frankensteining in our lab or in other people's labs. But it's really fascinating, the potential for research there. That's pretty neat. Dr. Harris, both of you attended the New York Stem Cell Foundation conference here in New York, right?
That's right. Yeah, the NICEF conference. It's an annual conference. One of my favorite conferences. I really enjoy attending. It was here in New York City at Hunter College, about 500 people from academia and industry, but from all different medical fields. So diabetes, cancer, you hear about how they're using the latest organoid and stem cell technology to address their own research. And so I find it very informative. Yeah, one of the most fascinating talks, though, was discussing organoid intelligence. And so organoid intelligence is the use of brain organoids as a tool for modeling higher level brain function, like learning and memory, right? So they talked about experiments where they connected neural networks, used neural networks like an organoid, and incorporated them into a simulated game environment and gave them real-time feedback so that the neural networks could learn. And the case that they showed where they were learning how to play a video game, Pong, which is a very simple video game. It's one of the first, if not the first. And so was fascinating that it could, learn how to play this video game. It's not that the organoids were thinking or sentient in any way. There was no memory of learning the game and so on, but it really showed how they could be valuable models in understanding brain function and a tool to understand how different genes or chemicals might interfere with brain function. So I thought that was really fascinating.
Yeah, that is the future. That's science fiction. How
Totally.
cool that you were able to go and see that presentation.
Mm.
We have an opportunity as a segue to give someone a little taste of science fiction here at the Tisch Lab. We're pleased to do a giveaway today and invite one participant to the Tisch MS Lab here in New York, where you can see Dr. Kalpana's research in person, look at the organoids, tour the lab, and then have lunch here at the center. So Dr. Harris, I'm going to ask you to pick a number between 1 and 100.
42.
42. Congratulations, Julie Polisina. Julie Polisina. Please reach out to us, marketing@imsmp.org, to arrange your tour and lunch. If you're not local to New York City, let me know anyways, and we will pick a different number and reach out to that person after the fact. But we would love to have you here, Julie, and we look forward to welcoming you to the lab. Now, we do have some questions that were submitted, and we're going to get to as many as we can. We had a few that were some variation of this: why organoids, why now? Why this, why now? Why is it important? Why are you doing a webinar on it? So Dr. Harris, why this, why now?
Yeah, I think brain organoid research is really exciting right now because it represents a major advancement in our ability to model the human brain in a dish in a way that was just impossible a decade ago. And we're already seeing results in diseases like MS. So it's really opening up new avenues of research to study disease. And I predict that they will become very powerful tools, both for, personalized drug treatment as well as for treatment development. So I think it's a topic everyone should know about because you're going to be hearing a lot about it in the future.
Yeah, it seems like it's revolutionizing the way that research is done. So to be at the forefront of it and have valuable contributions just speaks to what we do here at Tisch, but also, yeah, just really exciting to be part of. Dr. Kalpana, when creating an organoid, does it matter if the blood that you draw is B-cell depleted? For context, a lot of people with MS undergo treatment that does deplete B cells to help manage their disease. So does that affect their ability to organoid or not to organoid?
Yeah, so thank you for the question. So I believe it should not affect the way the organoid is developed. The organoid, because it is using higher order cells like neurons and other glia cells, and B cells yet are not really part of these organoid models that we're using. So I believe having a sample depleted of B cells should not affect how the organoid would be created.
Great, thank you. Dr. Harris, we had a question from a former patient regarding a bone marrow sample that is stored at IMSMP. Can their bone marrow sample be used to make an organoid?
Oh, interesting question. Yeah, I mean, we have, as many of you know, we have bone marrow MSCs, mesenchymal stem cells, that are banked, that we've taken from many patients, some of whom are participating today. And as part of the cell therapy clinical trial that's been ongoing at Tisch. And I think it's technically possible to take an MSC, it's an adult cell, and reprogram into an iPS cell and then make an organoid from that. I think that's probably technically possible, but it's probably more efficient. And most of the protocols for reprogramming have been based on using blood or skin as a starting material. So So I think it's probably more efficient to start with blood or skin samples. And I'll just say that reprogramming a cell and making a personalized organoid is something that is performed under regulatory oversight and informed consent. So no one should be making your organoid without you knowing about it.
Yeah, that makes sense. Given that MS can be so individualized in its symptoms and progression, how can organoids help in treatment development? And the second part of this question was, how do you balance scale for research and testing with individual application?
Yeah, that's a great question. Brain organoids are very powerful tools to model these differences in disease symptoms, in terms of progression, because they are personalized. And I showed you some of those differences in the paper, primary progressive patients. So in terms of development of neurons and oligodendrocytes. So we could potentially use these organoids to screen for new drugs that might reverse that phenotype, for example. So currently the technology, so I do think it will be very helpful in treatment development in that context. But the technology is really scaled for research at this point. And, I think the discoveries in the next 10 years will lead to a point where they will be useful for an individual application, but currently really mostly in the research realm.
That makes sense. And we've had a few other questions that I can see coming in through the Q&A feature, and we had others that were submitted through our form that were similar, like how can I sign up or can I participate? So I do just want to reiterate once more, correct me if I'm wrong, that organoids are a research tool. They are not yet a treatment. They are helping us find really effective treatments, but they are not yet a treatment in and of themselves. Is that correct?
Correct, not yet. We're years away from it being any kind of treatment, but a valuable tool for research. Yes.
Yeah, it's still really exciting because the treatments we develop are only as good as the information that we have. So if we can develop a better way of gathering information, then of course the treatments that we create off of those are going to be more precise and effective. Thank you. One more question for you, Dr. Harris. How would you advise undergraduate students to break into research regarding mechanisms of MS for potential treatment development? Very enterprising question that was submitted to us.
Yeah, as an undergraduate, you should really, if you're interested in research, particular MS research, really seek out labs at either your undergraduate institution or elsewhere that might have research opportunities, either during the summer or during the school year. Contact as many people as you can. Contact them early. Don't wait until Memorial Day to ask about a summer internship. I will plug that we do have a research assistant program where we hire recent college graduates. They work in our lab for 2 years as 2 gap years before going on to medical school or graduate school. So that's another great way to participate. There's usually information on our website about applying. So if you know anyone interested in doing that, please, check out our website.
I'm glad that you plugged it because I would have if you didn't. All of our research assistants are always accepted into the graduate or medical school program of their choice. They do really well here. They have the opportunity to present at conferences, get their name on papers. It's a pretty cool opportunity. Dr. Kalpana, why would the use of organoids as a model for experiments investigating MS treatments be different or better than using rats as models? I know you addressed this in the beginning, but can you refresh our memory?
Yes, of course. So as I mentioned in the beginning, in the presentation, like we do use animal models not for MS research. There are rats and mice that can be used for as animal models for MS research. However, multiple sclerosis is a human-centric disease, and a lot of the symptoms are faced by humans, us as humans. So the animal models like rats do not fully recapitulate what would happen and some of these human-centric and human-specific effects that multiple sclerosis would have on us. So organoid models being personalized to an individual make them much better models to do MS research as compared to animal models like rats.
Makes sense. We had another question submitted. Could this be a treatment for PPMS?
So as Dr. Harris just mentioned, and so did you, that we are far from a treatment option using organoid models. Organoid models are great research tools for us to understand how the disease progresses. The MS, as you know, is a progressive disease. So to understand the different aspects of this disease progression and to understand the interplay of the different cells that are involved in MS, organoids are great tools for that. They are still not a treatment option. Maybe in the future they might be, but as of now, these are just research tools for getting to that treatment.
Thank you. And powerful tools. The study that Dr. Harris walked us through earlier showed for PPMS specifically that different genetic component that we were able to discover. That wasn't known before about PPMS. So I can already see how they're shaping therapeutic creation. Dr. Harris, I have a question for you that's fun. And to the person that submitted this, I promise you are not the only one thinking it. So they said, as a writer, my mind goes there. Can this lead to a potential disaster where the organoids over time take over the organs, i.e. the human race?
That sounds like the beginnings of a really great sci-fi novel. So we should write it. I'd love to consult on that. Something about organoids just lends itself to like thinking about these like crazy outcomes. Yeah, that's still science fiction at this point.
Great, good to know. It's very reassuring. Dr. Kalpana, would the organoids that are developed work for a person who is 60 years old and was diagnosed with PPMS at age 54? Would I be considered aged out if this potential therapy does come to fruition?
So I would just like to reiterate once again that this is not really a treatment option. But having said that, yes, iPSCs from any individual can be used for making organoids. So no, you're not aged out, and then we can definitely make organoids from different individuals, including somebody who has had PPMS, I believe, at age 54 and are now at 60 years of age.
That almost seems to me like even more of a reason to utilize an individualized organoid model, because you could have one treatment that might work differently for someone of advanced age, or say someone that's 75 rather than someone that's 15. We'd be able to see how they would specifically react to a given treatment by testing it on their organoid. So not aged out, and perhaps an even better tool for someone that might not be at the median age of people that are diagnosed with MS. Dr. Harris, we had a series of questions and we've sort of covered this, but they asked a few particulars about like organoid results, side effects of organoids, reinsertion after growth. I think we've come to the understanding that organoids are not organ replacements. They're the way that we create bespoke treatments. But would you like to add any further clarification there?
Yeah. I mean, we're currently, as I mentioned, we're using organoids as a model to study disease and to screen for new treatments. And this is, where this field is now. So not necessarily organ replacements for the some of the reasons you mentioned. They probably won't be replacing organs, but rather in the future, in the far future, maybe repairing damaged organs. So I could see that in the distant future. I think I like to think about the safety of this approach in terms of a regenerative medicine. The side effects would be unknown, could possibly include cancer growth. So we would have to do a lot of clinical trials and safety testing in order to get to that point. We would somehow have to encourage the organoid when it's transplanted to get blood vessels to get in there so that they can get a blood supply after transplantation. So that is one important hurdle. Currently, we can't really scale and manufacture organoids to the point where they could be used as a therapy. But again, all of these things will be happening in the decade to come. So I think organoids as a drug screening tool is in the near future, but as a therapy, we're looking at many years away.
Got it. This dovetails nicely into a question we received just in the past hour. Will the Tisch MS Center conduct clinical trials using organoids? It seems like lots of clinical trials are going to be happening here at other labs, et cetera. Is that right?
Yeah, you know us. If we get to that point, you know that we will participate in trials like that. Yes, of course.
That makes sense. Dr. Harris, you lead a lot of our stem cell work, so perhaps you can answer another question that came in. Are organoids used instead of autologous stem cells? And perhaps we're talking about stem cell therapy versus using an organoid to decide on a treatment option. I'm not quite sure what this question is, but seeing as you do the stem cell work, maybe you have a better idea.
Yeah, I mean, one of the reasons we started the cell therapy program here using autologous bone marrow mesenchymal stem cells is because we could make them autologous. Transplanting any kind of cell that's not autologous would require immunosuppression potentially, and we didn't feel that would be compatible with MS. So at least the type of immunosuppression that would be required. So we've always really been more focused on autologous. Now with with IPS technology, we are exploring using IPS as an autologous starting point to either make MSCs or make other types of neural cells that could be tested in a clinical trial. So that is ongoing. Then to take that to the organoid level, I think, again, is many years away, but yes, in theory, it could be used as an autologous treatment.
Thank you. And I want to take a moment just to thank our participants today. Thank you to everyone came in. Thank you for your research, your interest in science, and your support of research. We do these things because we don't think that patients or their community or the public should be separate from the work that's being done in the lab. And just because Dr. Harris and Dr. Kalpana are wearing the white coats doesn't mean that they are the only ones that know this information. So something that I love about being part of this organization is that we trust our patient population with more info than you'd get at your normal run-of-the-mill neurology office. So thank you to everyone that's attended today. Thank you to Dr. Harris and Dr. Kalpana. We are out of time, but I'd love for everyone to mark their calendars for September 12th. That will be our next quarterly Connect webinar. You can expect an email from us about it, as well as an email on Monday with the recording of this session. And you're welcome to follow us on social media at TISCHMS and at imsmp.nyc. That's where you can keep up to date with behind the scenes information, as well as constant updates from our lab. We're really committed to providing you with info-rich content across our social media platforms. So again, thank you to the researchers for joining us today. Thank you to all of our participants, and have a wonderful weekend. Bye-bye.
Thank you. Bye.